As a very general rule, there are three types of information that are encoded in genomes. Probably the one that most people are familiar with are genes that encode proteins. But in the human genome, this represents only 5%, or even a little less of the entire genome. There are also regions, and nobody really knows at present exactly where they all are and how big they are. But we know very well they exist, and we've identified many of them that control when genes are turned on or off and how much. These are so-called regulatory regions. There are two different parts of the regulator regions. There are promoters that are located just before a particular gene that regulate gene transcription, and there are other more mysterious parts that are called enhancers, located far away from the gene in question. It turns out that the enhancers, which we think of as being very far away from the gene in question, actually may not be so far away. That's because up to now, we've been studying DNA and genomics in 2 dimensions. But life is in 3 dimensions. For a molecule of DNA, we can stretch it out, measure it, and read it in the ways that the modern technology allow us to do. But that's not how it works in the body. That's not how it works inside the cell. The DNA molecules are coiled, and when you look at 3-dimensional aspects of DNA, you sometimes find that these enhancers that previously, mysteriously seemed to be very far away from the gene they are regulating, are actually quite close by. And that's one of the really exciting new areas, studying DNA in 3 dimensions, and when you include a time component to that, actually 4 dimensions. So, a whole new science of 4D genomics is taking shape to understand the physical structure of a genome, and the temporal aspects under which it functions. The third type of important element that you find also in all organisms are regions, genes that encode RNA molecules. And RNA molecules are also essential components of every cell. Another part has to do with parts of the genome that aren't regulatory, and that are not genes, and that are actually relics of genes past. There's a lot of stretches of the genome that don't currently or produce any functional proteins. But yet it's still part of a long stretch, and it still gets copied and passed down from generation to generation. A lot of this has been referred to as junk DNA, and there's a bit of debate about whether it's truly junk. One of the best explanations about it was that, don't confuse junk with trash. Trash is useless, you throw away. Junk is what you keep in your garage because it might one day be useful or used to be useful, and then it broke, but it's still there so you keep it. You pass it on. So, there's a difference between junk and trash. We think that maybe large parts of the genome, which don't have what we think of as functional genes in them, had been there because large parts of the genome can get copied and put in a different location. So, if we think about genes as reflecting the genome as a recipe book, as an instruction book, there's a whole other side to the genome, and that is the genome as a history book reflecting the evolutionary history of that particular species. This has been a marvelous area of discovery leading to all sorts of insights, especially, but not limited to our understanding of human evolution. We can read the genome as a history book, looking at these parts of the genome that aren't actually genes at this point in time.